Welcome to this explainer on the complex endocrinology behind what a lot of people are calling nature's Ozempic. Today, we're exploring endogenous GLP-1 secretion via microbiome-mediated dietary fiber fermentation. Now, you've definitely heard of GLP-1 in the context of modern weight loss drugs, right? But the biological architecture to produce this incredibly powerful metabolic hormone already exists entirely within your own gut. We're taking a deep, evidence-based look at exactly how to activate it.
Today, we're going to track a fiber molecule's journey from the moment of ingestion all the way to its systemic metabolic effects. We'll be covering the enteroendocrine incretin system, mechanisms of colonic fermentation, keystone gut microbiota taxa, evaluating specific dietary fibers, synergistic macronutrients and phytochemicals, and finally, clinical protocols and tolerability.
Section 1. The enteroendocrine incretin system, understanding the body's largest endocrine network.
To kick things off, we start in the gastrointestinal tract, which honestly operates as a massive endocrine organ regulating postprandial glucose and centralized appetite signaling. Naturally stimulating your own pulsatile GLP-1 actually mitigates the severe gastrointestinal side effects we so commonly see with synthetic drugs. You see, endogenous GLP-1 has a transient 1 to 2 minute half-life, localized enteric signaling, and is highly tolerable. Contrast that with pharmacological GLP-1 agonists. Those are engineered for continuous receptor saturation, systemic circulation, and engineered DPP-4 resistance, which is exactly what leads to that high incidence of nausea and gastroparesis.
So to get that highly tolerable natural response, we have to look at the source, enteroendocrine L cells. These are specialized epithelial nutrient sensors, and they are the fundamental biological factories we're targeting to naturally orchestrate metabolic restructuring and insulin sensitivity. They're responsible for the synthesis and secretion of incretin hormones like GLP-1 and PYY. But here's the kicker. They are heavily concentrated deep down in the distal ileum and the colon, and that deep location is a massive clue as to how we trigger them.
Section 2. Mechanisms of colonic fermentation. How dietary fiber becomes a metabolic signal.
What's really fascinating here is that fiber itself doesn't actually trigger GLP-1. Instead, it's what the fiber becomes in the colon. Because those L cells are located so far down the digestive tract, simple carbohydrates never even reach them. We need complex substrates. And that brings us to short-chain fatty acids, or SCFAs. These are highly potent signaling metabolites, predominantly acetate, propionate, and butyrate, produced by the commensal microbial fermentation of non-digestible carbohydrates. Basically, your microbiome eats the fiber and ferments it into these fatty acids, which then bind directly to FFAR2 and FFAR3 receptors on the L cell membrane to drive the transcription of the pro-glucagon gene.
Let's walk through how this builds into a chronological, sustained physiological response. At hour 0, you ingest complex dietary fiber. By hours 2 to 4, that fiber escapes digestion and arrives in the distal colon. From hours 4 to 12, microbial fermentation produces continuous yields of those SCFAs. And then the magic happens between 12 and 24 hours, a sustained day-long elevation of your basal GLP-1 tone. You aren't just getting a post-meal spike, you are fundamentally shifting your entire biological baseline for the day.
Section 3. Keystone gut microbiota taxa, the critical biological middlemen.
As we've established, dietary fiber is completely inert to the human organism, without the specific ecological networks of bacteria required to ferment it. You need the biological middlemen. Enter Faecalibacterium prausnitzii. This highly abundant, gram-positive anaerobic bacterium is recognized as a paramount producer of colonic butyrate. Cultivating this specific butyrate engine actively protects your systemic insulin sensitivity because it's absolutely critical for upregulating colonic GLP-1 and PYY gene expression. It's a true hallmark of a metabolically healthy gut.
And then we have Akkermansia muciniphila. This mucin-degrading bacterial specialist is a monumental breakthrough in endocrine research. It proved that our microbiome secretes specific proteins that act precisely like hormonal master switches. Unlike other bacteria that rely on fermentation, Akkermansia secretes the P9 protein, which binds directly to your host ICAM2 receptors to drive explosive, SCFA-independent GLP-1 release. It completely bypasses the short-chain fatty acid pathway. How cool is that?
Section 4. Evaluating specific dietary fibers, maximizing incretins while minimizing gas.
Now, for someone sensitive to gas and bloating,
A high fermentation at any cost strategy is definitely the wrong approach. We need to evaluate the optimal tools. Type 3 resistant starch gives you intermediate sustained kinetics with high clinical tolerability for microbiome-dependent fat loss. Acacia gum is slow, deliberate, and very highly tolerable, a gentle bifidogenic prebiotic. PHG is slow and low viscosity, making it incredibly IBS-friendly while still yielding those great SCFs with low bloating. And beta-glucan provides sustained viscosity for delayed gastric emptying and even L-cell hyperplasia. These slow, deliberate fermenters prevent massive localized gas accumulation.
Conversely, you must strictly avoid rapidly fermenting fibers. Inulin has incredibly poor clinical tolerability because its rapid proximal fermentation just overwhelms the colonic mucosa. FOS and GOS are similarly poor, causing explosive hydrogen and methane gas. Even raw type 2 resistant starch can cause moderate to poor tolerability due to highly localized processing. So, if you're prone to abdominal distress and you see inulin prominently on a label, you're going to want to steer clear. Let's take a quick note on psyllium husk. It acts as a strictly mechanical adjunct. It's highly viscous but minimally fermented by your microbiota, meaning it does not directly stimulate massive GLP-1 secretion. But it flattens postprandial glucose excursions via absorption delay, and it elegantly mitigates gastroparesis and constipation without producing gas. It's a gold standard supporting player.
Section 5. Synergistic macronutrients and phytochemicals, amplifying the endocrine yield. We can exponentially increase our incretin yield by moving beyond isolated fiber and looking at complex nutritional synergy. For example, the Baobab protocol combines soluble prebiotics with bioactive polyphenols. Those flavonols and proanthocyanidins actually act as natural DPP-4 modulators, which extends the systemic half-life of your endogenously produced GLP-1. Add in some whey protein pre-meals to provide intracellular amino acid sensing, plus monounsaturated fats to simultaneously activate FFAR1 and GPR40 receptors, and you are literally conducting a prebiotic symphony.
Section 6. Clinical protocols and tolerability. Safe physiological implementation.
Knowing the science is only half the battle. To successfully harness this profound metabolic power without inducing debilitating bloating, we must follow strict titration protocols. Step 1. Low initial dosing. We're talking a baseline of 5 to 10 grams daily. Step 2. Gradual titration. Escalate slowly by just 2 to 3 grams a week. Step 3. Split your daily dose across 2 to 3 mixed macronutrient meals. And Step 4. Mandatory hydration. You must radically increase your water intake to prevent impaction. Your microbiome absolutely needs this distinct adaptation window.
Because listen, a fiber that is theoretically optimal but causes painful gas is practically useless. Clinical success is entirely dictated by adherence to a gentle metabolic ramping process. If you go for forced maximization and overwhelm your microbial capacity, you'll get painful gas and immediate abandonment of the protocol. But tolerable adaptation, that leads to modest, gradual weight management, sustained satiety, and localized tissue healing.
So what are we looking at for results? The likely payoff is a modest but incredibly meaningful improvement in satiety, postprandial glucose, lipid markers, and long-term weight management. This biological cascade won't replicate a synthetic drug overnight for sure, but it builds a deeply resilient, sustainable metabolic foundation.
Which leaves us with this final thought. Is the most powerful metabolic pharmacy already inside your gut? Armed with this highly structured biological framework, it really does seem like optimizing your microbiome is the ultimate key to mastering your own endocrinology. Thank you so much for joining me on this explainer, and I hope this changes how you view your gut health.